Good morning, everyone. So let's get started with questions. Any doubts, clarifications? None? So let's go ahead. Once again, a brief introduction to what is multidimensional NMR, kind of a block diagram of it. We realize that the 1D NMR will not suffice due to poor resolution. Meaning you will have too many peaks, and peaks also get broader as the size increases, due to which the resolution decreases. Therefore, there's a need for multidimensional NMR experiments. And how do we end up achieving these multidimensional NMR experiments? Is that we quite commonly exploit the one bond scalar coupling between proton and a heteronuclei, such as carbon or nitrogen, and end up doing polarization transfer or coherence transfer in order to increase the sensitivity of the carbon and nitrogen via the population from proton, and therefore get the frequency label and then detect it back in the proton. Why do we start and detect in proton? Because the sensitivity is proportional to the detected excited power 1 and detected power 3/2 for the respective gamma, and therefore it helps in getting better sensitivity. This was a pretext in which we said we'll predominantly do experiments that start from proton, go to a heteronuclear, finish in proton. In the meantime, collect chemical shift of the experiment. This way, our final density matrix will be of the form exp I omega x T1, product exp I omega h T2. This is a simple 2D experiment I'm demonstrating, which when you Fourier transform across T1 and T2, you're going to get a delta function at omega x convoluted by a delta function at the respective proton chemical shift. This way, a heteronuclear NMR experiment could be established so that you have a correlation between a necessary spin system, meaning the CH or N spin system, that can be studied within a given molecule, right? So these are general concepts. These are not restricted to biomolecules, but application we will be seeing predominantly in terms of biomolecules, because we said we are trying to understand a protein structure, and the basic structure or the monomeric unit is given this way, where the polymer is an extension of the amino acid. This is an example. This is a general structure of amino acid, where if R is equal to H, it becomes glycine. R is equal to CH3, it becomes alanine. And for instance, R equal to a phenyl ring, this will be phenylalanine. Sorry, CH2PH will be phenylalanine. R equal to CH2AR para OH, that will be tyrosine, and so on. Also other ones, I'm just giving some example of aliphatic and aromatic amino acids. And of course, you have tryptophan, you have charged ones like glutamate, aspartate. These are negatively charged. The positively charged one like arginine, lysine, and histidine, which could change its protonation state and could be characterized as aromatic and or charged, right? So you have a wide variety which are determined by the side chain group that is being present. The most fortunate aspect that we have here is that we have heteronuclei that are directly bonded to a proton. So in this aspect, we are fortunate enough to start with the nitrogen proton HSQC experiment, which forms the fingerprint for that given protein. NH15N 1H HSQC is a fingerprint. This is an example HSQC of the protein ubiquitin, and where you are able to see the nitrogen proton correlation. And this is a 76 residue protein. If you sit and count, you will come close to 70 to 80 peaks, which indicates for every NH that's being present, you will get one peak. And in addition, wherever you have a CONH2, you're going to get a bunch of 2 protons associated with 1 nitrogen. So all this end up arising from a CONH2 group that's present in glutamine and asparagine, which will have a CONH2 side chain that is being present. Okay, so that's why I didn't say 76 or above 80, because depending upon how many side chains, you're going to count more number of peaks. If you end up eliminating these, then you would find 1 cross peak for every NH in the sequence except for proline, because proline does not have an NH. So therefore, this ends up being a characteristic fingerprint for every protein. So this is the HSQC that is extremely helpful, right? So similarly, one could also exploit the CH that's being present, which will provide an idea of the backbone as well. But there's a small complication here, because you have R groups that are also other aliphatic groups. You're not going to cleanly get the C alpha H alpha. You're potentially going to get the C beta H beta. For alanine, it's a CH3 group. So you have C beta 3 times H beta, so which is going to be very intense peaks, right? So which is also what we end up seeing in the 2D. Let me see the 2D. If you see the 2D here, the methyl groups end up coming here. They are quite sensitive, and you will count more than the number of CHs for alpha, because C beta H beta, and there are some that have C gamma H gamma. There are some that have C delta H delta, and that some also have, if I'm not wrong, C epsilon H epsilon. But predominantly, these CH spins also end up coming in the same region. Therefore, the CH, although it's extremely useful, is quite still crowded to get your information. Did that make sense? Right? So therefore, the CH is, although useful, the most predominant one that's used as a fingerprint is the NH spectrum. Everybody comfortable so far? So therefore, if you want to assign the polymer, which has multiple repeating units of the same monomer, you have, for instance, this is a 55 amino acid protein. You have about 55 peaks, but I'm not sure this peak belongs to which residue. In order to do this, we need to achieve something called sequential assignment. What is sequential assignment? Sequential assignment is one where you are able to pick a peak and say that belongs to, say, residue number 3. And another peak belongs to residue number 22. And where do they end up coming in the HSQC spectrum? How would we assign what is 3, what is 22? Is when 3-dimensional NMR experiments are going to be extremely useful. The basic block diagram for a 2-dimensional NMR experiment is preparation, then T1 labeling, mixing, and then detection. What do we mean by this? You can go back and recollect what we did for the HSQC. The HSQC, the preparation phase is the INEPT part, right? Meaning that you started from HZ and you are able to successfully prepare a HZ-NY state. Okay, this is preparing the system in the coherence that you want it to be prepared in. Make sense, right? And then you have T1 labeling. How is T1 labeling achieved? Simple. You let it evolve for a period T1 by applying a 180 pulse in the proton at T1/2. This helps you achieve frequency labeling of the nitrogen in the T1 time domain, right? And then you mix it back and get it, for instance, and then you detect. This is the entire pulse program, right? If you have to think of the same thing for the COSY experiment, the COSY experiment is even more simpler, the proton-proton homonuclear COSY experiment. Here, what happens is that you prepare the system with a 90-degree pulse. You do T1 labeling. What does T1 labeling help in? T1 labeling helps in creating chemical shift for I, chemical shift for S, at the same time end up mixing I and S due to scalar coupling. Okay. Of course, if you started from IZ, you would have generated something like 2IXSZ from it. Go back to the density matrix, you will recollect this. So what you're able to see during the T1 delay itself, you're doing frequency labeling for the chemical shift, and the mixing also happens, right? And before the actual mixing is accomplished, when you end up generating something of this kind. Basically, you end up switching it back to the proton, or rather the coupled proton. You started from IZ and you finish in SY, and therefore you have prepared and you end up detecting. So what you're able to realize is that multi-dimension NMR experiments is preparing something, collecting some information you want and pushing it back and detecting, right? In the similar vein, when you're trying to talk about an amino acid, which is going to have the backbone of this kind, what you would prefer to do is to make sure, since this is your fingerprint, you pivot around the NH, meaning that if you're able to assign the NH, you'll be able to assign everything, meaning that you can go from here to here, therefore assign the C alpha. The moment you assign the C alpha, the H alpha can be assigned in the proton carbon HSQC, right? The moment you assign the H alpha and C alpha, you could do cozy type experiments to jump from H alpha to the side chain and get their assignments. Did that make sense? Let me repeat again. If you pivot, if you are able to assign the nitrogen proton HSQC, then all the other assignments could be gotten from the other 3D experiments by transferring from H to N and N to C alpha. The moment you get C alpha, let's just try to draw one extended chain. Say, let's say C beta, C gamma, there's a H beta, there's H gamma. Let's say these spins are there. The moment you go from H to N and N to C alpha and somehow get the sequential C alpha assignments, then you also get the H alpha. From H alpha, you can also always jump to the H beta through the cozy experiment, right? From H beta, you can always jump to the H gamma. Okay, with this, you'll be able to assign everything in a given protein except for the oxygen, because the oxygen is not NMR active. Make sense? So that will form the premise of today's class, where we will be looking at some of the three-dimensional NMR experiments and trying to highlight the different concepts that we have learned. And what are the concepts that we have learned? The first part would be transferring from H to N INEPT. So let's try to take the simpler experiment where we look at the HNCO. We take a look at the HNCO experiment rather. Right? How does the HNCO work? The HNCO ends up transferring from H to N, N to C, get this labeled, and then move back and go to the proton. So I told you yesterday there are two ways of doing this experiment. You can move from H to N and do nitrogen labeling, right? And in some ways transfer to the NC by an INEPT. Correct? Then you can do carbon labeling in T2, right? And then you can do reverse INEPT, C to N. And the moment you do C to N, the next has to be N to H, and then you detect in proton. Right? So you have T1 labeling as nitrogen, T2 labeling as carbon, T3 labeling as the proton. You started in the proton, moved to nitrogen, did nitrogen labeling in T1, then moved to the carbonyl carbon, did frequency labeling in that, jump back to get it done. These are reverse INEPTs. This is one way of doing it, right? Alternatively, what you can do, which I'm not going to write, is to go from the proton to nitrogen and the nitrogen to carbonyl carbon, and then do carbon labeling in the T1 dimension, push it back to nitrogen, do the T2 dimension, and then finish off on T3. Okay, let's try to take a look. Yes. Yeah, you want me to repeat the second one? In the first one, the approach A, omega S in T1, omega N in T1, omega carbonyl in T2, omega HN in T3. Correct? Alternatively, instead of doing this, you could also consider something like this, alternate route B, where the carbonyl is labeled in T1, nitrogen in T2, and of course the HN proton in T3. This will not change. but you can always swap the order here. Okay. What I'm trying to make you understand is that moment you have the building blocks, you can build the blocks based on the necessity that you want. Predominantly, we would take the route A because we already have a high resolution proton nitrogen correlation from the HSQC. The HSQC already gives you a very high resolution. What do we mean by high resolution? The T1 max, the acquisition time on the 15N dimension determines resolution in the indirect dimension, right? Between 2D and 3D experiments, which experiment would you prefer to have a high nitrogen resolution, 2D or 3D? Sorry, why? You said 2D, but why? What's that? Exactly. Time is the biggest deal. You could always get the same nitrogen resolution in the 3-dimensional experiment as well, but that's going to take a very, very long time. In quiz 2, you solved a problem where from 1D to 2D takes time. 3D will take even more time, right? For 2D, you needed to do at least 2 scans to get the indirect dimension frequency properly determined. Here, you would need 4 such scans. So you're going to spend more and more time to get the 3-dimensional experiment. So the easiest experiment to get a high resolution nitrogen proton correlation is the HSQC. You can go longer and get it done in the 2D fashion. So in the 3D fashion, since you anyway have a high resolution connecting between them, people tend to prefer the nitrogen to be labeled in T1 and carbon to be in T2. There's also another reason for it. Remember, the carbonyl carbon is kind of fixed in this given angle. The only way the chemical shift would be different is based on the C alpha CO dihedral. Of course, there are 2 Ramachandran angles. One of the Ramachandran angles will have C alpha and CO as the middle bond. There are 3 bonds that define a dihedral angle. Is that correct? So what will be that? N, C alpha CO, and CN again. So that is the dihedral we are trying to talk about. Okay, so carbonyl, since it doesn't have much features, is also not going to have a very good chemical shift resolution, right? Because they're going to be more crowded on top of each other. So it's also a smart thing to invest on the higher resolution on carbon in this experiment than the nitrogen. Does that make sense? Yeah, so now let's take a look at actual experiment. I'm taking the illustration from the Kavanagh book, so please go back and take a look at it. I'm also adding the figure number in it, 7.4. So this will help you understand and go back and read as required. So what are we trying to do here? We are starting from the proton, and I'm sure you guys are able to appreciate that is the INEPT module, right? And the moment you have the INEPT module done, you have generated something like 2HX and Y, depending upon the phase of phi1. The phase of phi1 is kept to X. So I am quite confident that you would have ended up generating something like at point A, 2HZ and Y. People who understood this, please raise your hand. Excellent. So now that is done. You are able to see the first thing that happens is T1 labeling, meaning that you get the chemical shift of the nitrogen encoded in the first time domain, T1 domain. Of course, you're only doing T1 by 2. Why? Because you have to apply a 180-degree pulse in order to decouple the proton from the nitrogen. And of course, you should also decouple the carbon. Remember, you're doing a HNCO experiment. And this is one point that I had highlighted yesterday. What was the point? It was that how can you have the same carbon channel divided into C alpha and CO? And in order to explain that, I showed you the experimental spectrum here, where you were able to realize that the C alpha comes between, say, 10 to 70 ppm. On the other hand, if you take a look at the carbonyl, it comes somewhere between 160 and 180 ppm. You're talking about 100 ppm difference in carbon. 100 ppm difference is going to be several kilohertz. Therefore, the pulses that you're applying might not be very effective. Reflect upon the tan theta argument that you did, where it's small omega 1 divided by capital omega. If the capital omega increases, off resonance effects increases. What do I mean by this? Is that you're talking about tan theta is omega 1 over capital omega, where the capital omega is the offset and small omega 1 is the power of the pulse. You guys might recollect it, where theta would be the B effective. Where does the B effective lie with respect to the z-axis, and how does precession happen with respect to it? If you're able to realize that the offset is very high, right, what is going to happen? If this value is a certain number and this value is very high, the entire value is going to be very small. The entire value is going to be very small, tan theta inverse, the theta will be very close to 0 degrees. So you can potentially apply a pulse in C alpha that will not affect CO. Similarly, you can potentially apply a pulse in CO that will not affect C alpha. So in theory, these two can be treated as separate channels by changing the carrier position, omega RFT. The omega RF of the B RF can be carefully changed to keep the carrier in carbonyl or C alpha to get the job done. Okay, so therefore, when you are doing the frequency labeling on nitrogen, you have to decouple the proton and the carbonyl. Why? Because while the nitrogen is being labeled, you have coupling that will arise, one bond coupling that will arise from the HNCO. Are you guys able to understand the fact that frequency labeling is happening on nitrogen? The proton and the carbonyl have to be decoupled. And how do we end up decoupling? Apply a 180-degree pulse in middle of T1/2. But this is a very interesting module. This module you guys have not seen before, right? Generally, what you would have expected, T1/2, 180, T1/2, with 180 being simultaneously applied, where this is T1 evolution. This module can be thought about as a constant time module. What is a constant time module? Is that if you take a sum of this, T1/2 plus capital T/2 plus capital T/2 minus T1/2. Do you guys realize that the T1/2 cancels? And you effectively have an evolution for a total period of T, irrespective of the value of T1, right? The advantage of this module is that in addition to getting the chemical shift evolution of nitrogen, it will also do the scalar coupling evolution of nitrogen to carbonyl in this duration. That's where the simultaneous 180 helps. Okay, and at the same time, this 180 will also help decouple the proton from nitrogen with respect in the T1 axis. My request to you guys is to go back and do the density matrix formalism starting from 2HZNY, and your active components in this period are, let me write it, active components during T1/2, T/2, 180, of course 180 on all the channels. Be careful about the phase, T/2 minus T1/2. During this duration, your Hamiltonian, active Hamiltonian will be nitrogen Nz, right? Pi J NH to HC NZ. And you will also have pi J NC prime to NZ C prime Z, where C prime is a carbon. So my request to you is that go and do this module, which will help you understand nitrogen chemical shift will evolve along with the N-C prime coupling that will also evolve. So basically you achieve two things simultaneously. You get the chemical shift labeled and you transfer the coherence to carbonyl. Can somebody tell me why are we trying to do two things at the same time? I'll tell you what this T should be set to in one moment. But why are we trying to do two things at the same time? Can anybody answer? Let's take the example of doing two things one after the other. You could have T1 labeling where you are T1/2, 180 T1/2. Then you could have had an INEPT to get it done. That is the easier way for us. You could have done the frequency labeling followed by INEPT so that you got the nitrogen chemical shift and moved to the carbonyl carbon. Why are we doing both together? Why are we in a hurry? Sorry? Excellent answer. To reduce the time. Why do we want to reduce time? (3D NMR takes long time, so you want to reduce time. That's a more important reason than that. Remember, you're talking about few milliseconds here. Your D1 time is 1 second. So cutting 10 milliseconds is not going to make a huge difference on 1000 milliseconds, right? So yeah, it's 1% change. Your answer is right. You want to reduce the time of evolution. Why? What is happening when this pulse program is implemented that we are not accounting for?
Relaxation.
Excellent, relaxation, right? So if you did T1 time followed by the INEPT time, and INEPT time is going to be 1 over 2J. And we just saw yesterday for the NC couplings, at best it's 15 Hertz, right? Therefore, if you have to set 1 over 2J, 1 over 2 times 30, right? Sorry, 1 over 2 times 15, which is 1 over 30, right? How much is going to be? 1000 over 30 milliseconds. So you're going to be talking about 33 milliseconds. That's a long time. And on top of it, you also have to do T1 labeling, which is another 10 milliseconds or so. You're wasting 40 milliseconds in this process. Since anyway, you have to spend the 33 milliseconds, now do you see the advantage for this constant time module? Until small T1 reaches 33 milliseconds, you will only spend 33 milliseconds to get both chemical shift of nitrogen and scalar coupling evolution of N to C prime. And we already would have gotten a high resolution of nitrogen from the HSQC. So probably the T1 that we'll try to go is maybe say 16 milliseconds, not more. You don't need anything more than that. So what we are able to achieve is that within this constant time module, we have chemical shift of nitrogen and the N to C prime INEPT transfer happening simultaneously. People who understood this aspect, please raise your hand. Okay, I see few people not raising hand. Let me repeat it again. So let's ask, in the nitrogen dimension, you might want the T1 to go, let's say, until 10 milliseconds at least. What is 10 milliseconds? The resolution will be 1 over 10 milliseconds, which is going to be 100 Hertz. This is very bad if you think about it. Of course, after zero filling, after other things, you'll get high resolution, right? But this is the best you could do, because 10 milliseconds with a spectral width of, say, 30 ppm, try to do it on, say, 700 megahertz on proton, you will realize that this itself is a lot of time. This itself will be, say 16 points in the T1 dimension. 16 points means you have to get 32 different experiments to get the nitrogen chemical shift because of quadrature, right? So that's going to take experiment time. That's one aspect which one of the person raised. But more importantly, if you want to transfer from N to C prime, you need to spend 1 over 2J for the INEPT delay. And the J for this is 15 Hertz for the N to C prime. Therefore, you got to spend 1 over 30 seconds, which is about 33 milliseconds. Both put together, you get 45 milliseconds or so, 43 milliseconds, but you're spending about 40 to 45 milliseconds in this duration. Somehow, if I can incorporate this T1 evolution within this, instead of spending 43 milliseconds, I'll only spend 33 milliseconds. I save 10 milliseconds. This will not help in the total measurement time, but this will help reduce the amount of relaxation that happens because you generated coherence along the transverse plane, which will undergo T2 relaxation. T2 relaxation will reduce your signal intensity. You don't want to reduce your signal intensity. So by combining this module, the constant time module, you're able to get the frequency labeling and have the INEPT transfer happen simultaneously. Excellent. Let's move forward. So far what we have done, do the INEPT transfer, do the frequency labeling for nitrogen, and do the NC prime transfer. Now, at this juncture, C, you are now generated 4 HZ, NZ, phi3. What is phi3? Phi3 is also set to X. So you generated something like CY. But remember, it also has a cos omega n T1 going with it, which is a cos modulation. Make sense? Now, the next part is obvious because we have seen it over and over again. This is where T2 evolution happens, which gives you the chemical shift labeling of the carbonyl carbon, right? With the two 180s on the proton and nitrogen, ensuring decoupling happening, okay? Right, this 180 is very interesting. Maybe you are thinking about 2 bond JNH to C prime coupling, but there are also other things that could happen. For instance, what we are trying to say, the coupling of this to this won't be very high, but still you're trying to decouple it so that you can keep your coherence for a longer amount of time. Any evolution that happens, it's going to cause relaxation. So you want to minimize such relaxation that comes up. So at the end of D, you would have gotten 4HZ, NZ, CY, cos omega n T1 cos omega C prime T2. So now at this point, what you're able to realize, this is the reverse inept for C prime back to N. And what happens here is the reverse inept for N to H. And therefore, you went from proton to nitrogen, got the nitrogen labeling, transferred it to carbonyl, got the carbonyl labeling, going back to nitrogen, going back to proton, and you're detecting. That is the out and back experiment that you're trying to look for. Now, you would only get a signal, of course, you're propagating it here, and therefore the final density matrix is going to have one more term in it, which will be exp I omega H T3, right? Direct dimension, you could get quadrature in one scan. Well, for the indirect dimension, you got to get quadrature from respective sampling of cos and the sine part. So this pulse phi1 will be cycled for quadrature. Similarly, this phi3 pulse will also be cycled for quadrature. So you need 2 scans for this, 2 scans for this. You need totally 4 number of scans to make sure in T1 and T2, you can get proper frequency labeling for nitrogen and carbon. So now you're able to see immediately from 1 scan experiment to get the 1D, you need 4 scans to get the 2D. You want 2 scans to get the 2D. Therefore, if you have a 4-dimensional NMR experiment, you need 8 scans to get the quadrature. For a 5-dimensional NMR experiment, you need 16 scans, assuming 1 scan is enough for sensitivity. So slowly you realize going from 1D to 2D through ND, you're going to require 2 power n number of scans to get the quadrature across every indirect dimension. So while you're able to get higher information by doing multidimensional NMR, adding more and more dimension comes at multiple costs. One cost is the total measurement time, which increases as 2 power n significantly. Secondly, where n is the number of dimensions. Secondly, you are also going to have multiple inept or polarization coherence transfers that have to be implemented. That is going to cost overall loss of coherence or, the magnetization that's going to reduce your overall sensitivity. So as you increase more and more dimension, it comes at a very good advantage of resolution, but at the cost of total measurement time and sensitivity. So unless there is a need, people will not keep adding more and more modules. Okay, so 3D is a very good optimum such that you get good resolution and reasonable sensitivity. So as you guys might remember from yesterday, we just discussed the pulse program of the HNCO experiment, which correlates the HN of a ith residue to the I-1 carbonyl. And if you set the sensitivity of that experiment to 100, you will see that as you go further in other 3D experiments, the sensitivity drops significantly, especially an experiment, something like CBCA CONH or CBCA NH as 1/100th of the sensitivity of the HNCO. So generally, if you take about 10 minutes to get the 2-dimensional HSQC for a protein, which is fully labeled, you would probably take 30 minutes for the HNCO. Probably you will take 12 to 24 hours for the CBCA NH experiment. We'll see what that experiment is if we have time. But overall, the idea is that you can keep building upon all of this, right? Okay, now that you've got an I to I-1, we could also exploit another experiment, which is the HNCACO experiment. What is this HNCACO experiment? HNCACO experiment correlates the HN of the ith residue to the ith carbonyl itself. Okay, and that's going to be an interesting experiment where, let me draw the the residue again. H, N, C alpha, H alpha R, C O. So this is the polymer. The previous experiment connected these. Now somehow, if you are able to connect this with this, you would have a common point which helps you on both sides. Therefore, if you ask for the same NH, who's my preceding, who's mine? And ask the same question here, who's my preceding, who's mine? Do you see that this will help you connect this nitrogen to this nitrogen? Let me repeat again. You want to get a sequential assignment for I plus 1 and I-1. In the HNCO experiment, you got on the N to I correlated with C prime of I minus 1. You got these two correlated. That is this experiment, right? Now, in the HNCACO, you're going to have Ni that correlates with C prime of I, meaning that you'll get these two correlated. Then why did we write this Ca in bracket? Is because you jump, previous experiment, you jump from H to N, get the T1 labeling, and N to C prime, got the T2 labeling, jump back to finish in proton as T3. You'll do something similar in this experiment. Instead of jumping from N to C, you will jump from H to N and N to C alpha. Remember, carbonyl chemical shift and C alpha chemical shift are well resolved. Therefore, you can easily jump from H to N and N to C alpha. So the way you jump from H to N and N to C prime, because this is around 170 ppm, this is around 50 ppm. Very well fall resolved. True? You can jump from here to here, here to here, and then you can jump from here to here. That's another one bond coupling, about 55 hertz or something. So this is about maybe 11 hertz or something like that. This is 11 and this is 7 if you remember. And this to this might be something like 55, which will be the review that we are trying to look for. There you go. Yeah, that's about 55 hertz, right? So what's going to happen? You're going to jump from 90 hertz. That will set the 1/2J delay. This one to this one is 11 hertz. This one to this one is 55 hertz, right? The important aspect of this is that although you jumped via the carbonyl, you will not do frequency labeling on this C alpha because that spends time on it, isn't it? Right? That is why it is written in bracket. It is still a three-dimensional NMR experiment similar to the HNCO. The only difference between the HNCO and the HNCACO with the CA in brackets is that it will not get the C alpha chemical shift, and it correlates the ith amide nitrogen to the ith carbonyl carbon. Right? So now what is going to happen for every I, you're going to get the I plus I minus 1 and I. And therefore, similarly, as much as you've gotten that for this one, you'll get the same thing for this one, isn't it? The same information will be applied for the next one. If you put all this together, can you stitch them together to get the entire assignment? At that moment, although you did not know which nitrogen belonged to what, you will be now clearly able to say which nitrogen belongs to who at that point of time. So this process of getting sequence-specific resonance assignment of a protein or any biomolecule, it's an important step towards characterizing that molecule. And therefore, your HSQC will now have labels in it. Meaning, what do I mean by that? Now, instead of trying to say, I do not know what this belongs to, I can potentially say it belongs to the 53rd amino acid. So, which is also one example that I show here. This is a very large protein where this belongs to valine, this belongs to a certain isoleucine, these two belong to cysteines, but these two are different cysteines. The residue number is written as 1827 and 1828. Okay? Does that make sense? Let me see whether I have any other assignments similarly. Yeah, so this is one example. Instead of protein, it's an example of a DNA duplex where every resonance. So for instance, you have G11 here, the G11 assignment is shown like this versus where G2 and G10 are in separate places. G2 and G10 are in separate, different places. Because this is sequence-specific assignment. You know which resonance belongs to which part of the biomolecule. This is important because then you can see the atomistic changes that happen in that given molecule when you add a drug and when you make it unfold, right? Protein-protein interactions, protein nucleic acid interactions, all that can be studied. So now the question that is being raised is that what if you're unlucky and have a situation where for two different residues, their amide proton, amide nitrogen, and carbonyl carbon have the same frequency by chance. The probability is low, but the overlaps do happen. They may not exactly be on top, But since we are not acquiring a very, very high resolution in the T1 and T2 dimension, you could have overlaps that start to come. Which is the proton, small differences proton will help you detect because you can get a high resolution proton, provided you have the sensitivity for it, right? This is also a place where, remember, we are not stuck with these three only nuclei. I'm happy that you raised it. Therefore, we can have better experiments. What are the better experiments? Not just correlating the HNCO, you could also correlate the HNCA, which was the example that I gave yesterday. Unlike the HNCO, which only correlated the I to I-1 carbonyl, the HNCA will correlate the ith nitrogen to Ca of I and Ca of I-1. The same experiment will give you both. Okay? You don't have to stop there. You can do another experiment, which will be HNCOCA. What is HNCOCA? HN. Go through the carbonyl and go to the previous C alpha. So in this experiment, you'll get N to I to Ca of I-1. So now what have you added? If you do the HNCO, you do the HNCACO, both put together should give you Hn of I, N of I, C prime of I with an information C prime of I-1 that should give you the sequential assignment. So you can have another pair of experiment, which is HNCA, which will give you an HNCA and HNCOCA, which when you repeat, you'll get Hn of I, N of I, C alpha of I and I-1. So now what you have done, you've expanded the entire set such that the N, Hn, C prime and C alpha for the I and I prime come, meaning that these are two different residues. It's very unlikely that literally all of them will stand on top of each other. You will get the better resolution. But you are still not wrong. As the protein size increases, overlaps start to increase. Oh, then what do we do? Well, we'll add one more sweet to this experiment. What is that experiment? HNCACB. Okay, let's take a look at what does a HNCACB do. We'll not look at the pulse program. We'll try to do it. Where is HNCACB? There you go. The HNCACB experiment is one that correlates the amide proton nitrogen to not just C alpha and C beta of the ith residue. It also correlates with the I-1. So now you're adding C beta into the mix. And remember, not all residues have C beta. For instance, glycine does not have one. The remaining 19 have. And remember, the C beta is directly attached to other functional groups, like phenylalanine, tyrosine. Therefore, the C beta chemicals could most likely be not degenerate between I and I prime, especially if they're not like similar amino acids. Okay, within the same lysine, you might have trouble. Oh, lysine has C beta, C gamma, many more coming up. So the C beta could still overlap. But it's okay. Let's divide and conquer. Let's get one by one done. But remember, this is only going to give you I and I-1. Therefore, you could also do another experiment. Now, what are we looking at? We are looking at the HNCACB, which gives you C alpha, C beta of I and I-1. You could do an experiment called CBCACO and H, which will give you HN and N with C alpha, C beta only of I-1. So therefore, slowly you have clubbed experiments like HNCO, HNCACO, HNCA, HNCOCA, CBCACONH and HNCACB to get I and I-1 for carbonyl carbon, alpha carbon, beta carbon. You don't have to stop there. You can extend this to also get the H alpha, H beta chemical ships, which are proton chemical ships. Slowly, you can devise experiments by building up on the different INEPT modules to get a lot of different chemical ships, HN, N, C prime, C alpha, C beta, H alpha, H beta, And depending upon C gamma, whatever, C gamma gets too much, then you have to have more transfers. So probably people stop right here. Now you have it for different residues going from the HN as your base. It's quite unlikely that all of them will fall on top of each other. For many nicely behaved proteins like ubiquitin, HNCO and HNCACO is enough, or HNCA and HNCOCA is enough to get your sequential assignments. But as the protein system starts to become bigger and bigger, you need to put a bunch of experiments to get all of these assignments to resolve from the degeneracy issue that you might end up facing. Okay. In this mix, while I was talking, silently I slided CBCA, Until now, all the experiments you looked at started with HN. Hey, what is this CBCA, And suddenly you started with CB. So this is a very interesting experiment where in the previous times, we always started with the amide proton, went to amide nitrogen, did whatever we wanted, come back and finish in the amide proton. Yesterday, one of the students raised the point, why should we always go and come back? Can we just finish in the nearby proton? That's the experiment of CBCA. CBCA experiment ends up exciting the alpha protons, which transfer the coherence to the respective carbon. And that ends up transferring the coherence to the nitrogen, which end up transferring to the proton and finish. This is still a proton to proton experiment, but this is not out and back. What is out and back? You give and take it back. Here, you don't give and take it back. Somebody gives, somebody takes it back. Right? Still proton to proton, still high sensitivity. True? Therefore, experiments could be designed in ways that are extremely convenient for us to maximize the sensitivity. You might always argue, why don't we go from here to here to here to here and then come back and do it? Immediately you see how many transfers we got to make. Right? On the other hand, a single transfer is enough to get it done. It's very similar to the fact of you guys buying a one-way ticket at times versus buying a two-way ticket at times. When you go on train, many times people train or flight, you buy a return ticket, right? Sometimes you don't have to buy it because you probably go for a long time and you want the convenience to be there. Similarly, you keep the convenience to yourself on whether to start from one, go and then finish at some point, or what you want to do, right? For instance, a HNCO experiment gives you the convenience of going back and forth because if you finish here, whom will you detect from? You have only a carbonyl carbon, direct detection on carbonyl carbon is going to hit you on the detected carbon, so that's not going to help. So you are better off coming back. On the other hand, the HNCACB, which is also the other experiment, HNCACB, that's a very interesting experiment where you would start from the HN, go to the CACB, and go look at the pulse program, whether you finish here or you finish here. Right? What I have done in this class is not to give you an entire idea of how does a protein structure come. But I hope if you know the primary sequence, which is what is drawn here in a schematic, if you do a suite of 2-dimensional and 3-dimensional NMR experiments, all these are 3D NMR experiments. There are more experiments. Let's not worry about these right now. You can have even more complications to get a lot more done. But would you guys agree, if you do all these experiments, at some point of time, all the resonances in the carbon and nitrogen will be assigned? Now, do you realize that is the fingerprint for your protein molecule, and any changes that you say, oh, no, actually only this residue reacted to the change while the other did not. That's the power of NMR. And you don't destroy the sample, right? You still see the system as it is. You see it at equilibrium, and you can actually study it near physiological conditions without any changes. The only disadvantage is that you need to have a lot of concentration. That's about it. Let me stop this lecture here. Tomorrow's lecture will be how to get the tertiary structure of the protein. Currently, what we have done is trying to say, okay, that's the polymer. You can get chemical shifts and say where the changes happen. But one of the important things is to say, okay, either it's an alpha helix or a beta sheet or a loop, right? That's a very important information to how do we get that will be the next question. Very important question. I'm sad that I kept that to the last part of this course, but that's an experiment called the nuclear Overhauser effect spectroscopy, which gives you approximate distances. That's what we will see for the next 2 classes. One more class revision, and we'll wrap up the course. All right. Thank you very much. See you guys tomorrow.